Okay, hi everyone. Welcome back. Hope everything is going well for you so far. We're delving into some really cool things, and we're also going to start integrating a lot of the material that we've been learning over the last two lectures in today's lecture. And it's actually a really fascinating thing. Have you ever sat and thought about how our brains work and why our brains are actually the most dominant brain on the face of the planet? And it turns out that there's a reason for why we're the best thinkers currently on this planet. It has a lot to do with some of the biomechanical features that we're going to talk about a little bit later on today. So with that said, I'm hoping that you're going to be really excited to learn why our brains and our neurons within our brains are so amazing, why they work so incredibly quickly, and why we can outthink an octopus or a squid. So as I get ready to start sharing my screen, just bear with me for 1 second. And we'll get right into things. So as I get into presentation mode, hopefully you'll be able to follow along with your notes. And how's that going for you, by the way? I'm hoping that it's all going well and that you are able to take down notes as you're kind of listening to this lecture. If I'm speaking too quickly, please let me know. We are on episode 3 or lecture number 3, and we are still in this neurophysiology section. And so we're going to be doing that for a couple more lectures before we get into some other really exciting aspects of the nervous system. All right, so let's just get going. So we're going to talk a lot about octopi today. And this octopus that you see down here at the bottom actually has a very interesting nervous system that we'll talk about a little bit later on. And we're going to talk about the speed of conduction. How do we get that action potential that we looked at in the last lecture? How do we get that from one end to the other as quickly as we possibly can? And in order for us to understand that, we need to look at what we call the excitability, the excitable properties of the cell, or the cable properties of the cell. And the two major factors that are going to play a role is myelination as well as axonal diameter. And this is going to be related to what we call the conduction velocity. These are really big words right now, but I'm hoping that as we go along in today's lecture that you'll be able to understand it. Just a little bit of a word of warning. Today it's a little bit longer than usual, so we have about 40 slides. And again, I'm hoping that you'll be able to take down notes as we go along, and we'll get all the way to the end of the Axon today, reviewing some concepts that we learned about in the last lecture related to kiss and run, full fusion, and we'll try to tie up some of those different ends. Sounds like a plan? I hope so. So let's just dive right in. So when we talk about impulse or action potential conduction, how the action potential moves when a patch or a section of excitable membrane, and remember what the definition of excitable membranes are, it has voltage-gated sodium channels. I knew you were going to say that, so I kind of beat you to the punch. So this causes that influx in sodium that we talked about in episode number 2, and this will reverse the potential difference, the positive charge and negative charge across the membrane. And this local reversal in the potential across this membrane temporarily goes from negative on the inside to being more positive on the inside. And we spent time last lecture looking at the roles of these voltage-gated sodium channels and voltage-gated potassium channels in being able to do so. So this local reversal in potential serves as the source for the depolarizing current for the adjacent membrane. So sodium channels, as you go further down the membrane, will also be opened. So as you get more positive influx, that charge spreads locally to the sides, and this will also carry that reversal in potential to the adjacent membrane as well. So once it gets started, this action potential will be able to propagate or move from where it started on the cell to the next patch of that cell. So again, remember, most cells have a concentration difference. Most cells, though, don't have the capacity to generate action potentials, and this is because they lack those voltage-gated sodium channels that we spent so much time on in the last lecture. They can conduct passive currents, but they won't generate action potentials. Very key difference, and one that you need to understand. So most cells are also not interested in carrying a signal any distance as a result. So think about these round cells that you might get within your skin or within other organs. They're not really interested in carrying a signal any distance because they don't need to communicate with each other, nor do they have this structure known as an axon. An axon is this structure that we find on neurons, which is a long extension of the cell body, and it looks very much like a wire, and we can actually think of that as a theoretical model for what this axon looks like, as a wire in terms of the capacity to carry electrical current. So it can carry that action potential down this wire, which we call an axon, to some other location at a distance from the cell body. So only neurons with these long axons and muscle cells can generate these propagating action potentials. So again, they're the only ones with these voltage-gated sodium channels, keeping that in mind. So here we are when we're looking at this neuron, and we look at the cell body, and this cell body does not have voltage-gated sodium channels. It's a membrane, but there are no voltage-gated sodium channels, and so we don't really get action potentials being generated here, but we have a large cluster of voltage-gated sodium channels at this structure right here called the axon hillock, or the initial segment of the axon hillock, and this is where we can generate action potentials. This is the first part of a neuron that can generate these action potentials. These other structures, like the dendrites as well as the cell body, are where we can integrate signals that are coming from other nearby neurons. And we'll come back to that in future episodes. So here's where we integrate signals, decide whether or not we are going to reach that threshold value that we talked about in the last lecture. If it reaches threshold and it's able to reach threshold at the axon hillock, here the voltage-gated sodium channels will open and it will become self-propagating. It will move down this particular axon where it will contact the next cell down because of the axonal output signal. And that's the majority of what we're going to be talking about later on today. All right, so just to organize you, we're going to be looking at how we can generate this action potential, how it carries down the length of this axon, what makes it so unique, how do we make it go faster, how do we make sure that it reaches its end as quickly as we can, and then what happens when it does reach the end, what happens here at these axonal terminals, which we call these presynaptic boutons or these presynaptic axonal terminals. So let's go back to these excitable cells. So in biological tissue, if we were to put voltage across this membrane, so we have this potential difference, remember, positive on the outside, negative on the inside, and we were to put this potential difference as square wave or a step change, and this is what it means. It looks like a stair or a step, and this step change in voltage that you see here across this membrane, it will try to traverse along the surface of this membrane. And we can measure the movement of this step potential change across this membrane in this direction, because we're looking at moving this signal. And if we were to measure it down here at the end of this particular patch of membrane, what we started with doesn't look anything like what we end up with. And in biological tissue, this voltage change that occurs across this membrane is something that we have to take into account. On the other hand, if we were to just use a wire, because that's the analogy that we're using, if we put in voltage on one end, it will travel down the length of this wire. And on the other end, on a copper wire, for example, instead of losing the signal as we would in biological tissue, we're left with exactly the same identical tissue. The same identical voltage that we started off with. And that's the difference between a biological system and the systems that we have within the neuron. And we need to understand that a little bit. These membrane properties, the things that actually make up the membrane, will actually alter the form or the shape of that input signal that we just looked at. So we're going to be losing signal. It gets smaller. It doesn't look like it's the same size as this current or as this potential travels along the membrane. So the key thing is, how do we prevent this loss, or how do we move this signal down this axon without it losing any of its strength? So you can imagine like trying to call someone, and if the wire connection isn't really good, it makes it hard to hear. You'd have to start shouting louder and louder to get your voice across. That's not something that we want in a biological system. And in order to understand how these things work and why we look at these different factors, we have to understand that when we model the axon, or we model these excitable membranes, we have to examine what we call a cable property. And to describe this property of how to get a signal from point A to point B without the loss of signal, we can define it by what we call the length constant of that particular membrane or that particular cell. And this is also known as lambda, or identified as lambda. And this lambda, or the length constant, measures how quickly the potential difference, what we start with on one end, will disappear as it traverses down the length of this membrane as a function of distance. Eventually, it will decay all the way down to 0. So this is the conduction velocity of the action potential, and we can measure it using this value known as the length constant, or lambda. It's a great place to take a little bit of a break to make sure that you're understanding these concepts before we kind of move on, because it is a really important concept. Again, we're only about a quarter of the way through the slides, so if you need to take a break at any time, just hit pause, come back whenever you feel comfortable. So if you're still with me, or if you're coming back, in terms of the cable properties, we need to understand, in terms of the nervous system, how do we make a signal go from point A to point B as quickly as we can without losing any of that signal. And there are two basic phenomena that we need to think about, and this is part of the cable properties within the nervous system, especially the mammalian nervous system. So lambda, this length constant, can actually be increased by increasing the diameter of the axon. So how wide the axon is will actually change this length constant. The larger the diameter of the axon, the less internal resistance there is, and as a result of the reduction in internal resistance, the less voltage is going to be lost across that resistance as that current, those ions travel down the length of that membrane. Additionally, we can also increase lambda by increasing the membrane resistance. The higher the membrane resistance is, the less current is going to be leaking out. So think of it like insulation. It's not a perfect model, but you can think of it that way. And more of the current will be forced down that particular membrane. An important slide for you to make sure that you know, and the next few slides actually illustrate this point in a little bit more detail. Mathematically, what I just showed you on that previous slide is all represented here on this particular slide. So the length constant of an axon that we're looking at, because that's the excitable membrane. Remember, we don't have that in the cell body. This length constant is defined with the internal resistance of the axon, the extracellular fluid resistance, as well as the membrane resistance, because that's where the signal is traveling, on the surface of the axon, because that's where we find the channels. So since the extracellular fluid resistance, we can't really change it. You can't really change the fluid that's sitting around your neurons, and it tends to be relatively low, like there's almost like no impediment to the movement of ions in the extracellular fluid. The resistance is almost negligible. So when we look at this particular equation, when we look at lambda being the square root of the membrane resistance over the extracellular resistance and the internal resistance of the axon, because the internal resistance is so much bigger, we can just cancel the RO out completely. And so for the most part, in the nervous system, we're only looking at membrane resistance and internal axonal resistance. And again, these are all concepts. If they're new for you, stop at any time, jot down some notes, come back to me with those questions, or put them on discussions, and I'll be happy to try to help as much as I can. So again, going back to the length constant, there's actually a way that we define this. So lambda, or this length constant of an axon, is defined as the distance that a potential or an electrical signal will travel to the point where that electrical signal drops to 37% of its original value. And so you can take a look. Here is 37%. We started off with 100%. And as we move, and let's say that this is at the very beginning, at the initial segment of the axon, and as we move further down the distance on that axon, at some point, if nothing else is going on, that signal will decay to 37% of what it started with. And that would be defined as the length constant of that neuron. And if we take a look, we're comparing here on this one diagram, we're actually looking at two different neurons, or two different axons of two different neurons. I'm hoping I'm not confusing you. I'm hoping it's clear. But again, any questions, jot them down. Happy to take them up with you later on. So we're looking at the membrane resistance of neuron or axon B. and we're looking at the membrane resistance of axon A or neuron A, and we're comparing the two. So sitting side by side, which one is going to have the bigger length constant for the same distance? And we can see that the membrane resistance of axon B is much, much larger. So it can travel a lot longer before the signal drops off versus neuron A, where it drops off much more quickly, and the lambda, or the space constant, or the length constant for neuron A is much shorter. So again, these length constants are a way for us to tell like how quickly an axon will conduct or move an action potential. And shown in a slightly different way, going back to that circuit model, if we were to put in to the membrane a certain amount of charge, 100% at this particular time, it wants to spread that out. It will lose that signal across the membrane. And as it loses it, it eventually will stop dropping lower and lower. And what we're measuring is how far will it have gone the length of that axon before it drops to 37% of its original value. So what are the things that can actually help with this length constant? So one way that we can do this is to increase the membrane resistance. And we can increase the membrane resistance by a process biologically known as myelination. And this is the most efficient means to increase this conduction velocity. And how do you myelinate? Well, we have non-neuronal cells, which we call glial cells, that can help out the neurons in the nervous system. These are used for nutrition. They're also used to increase the membrane resistance. And very specialized types of these glial cells, known as Schwann cells in the peripheral nervous system, or PNS, or oligodendrocytes within the central nervous system, or CNS, wrap around successive sections of the axon, and they will produce this covering known as myelin, or they will form what is known as the myelin sheath. All right, sounds good so far, hopefully. So let's take a look and see how this might happen. So here we're actually looking at two different cells, and we're looking at two different cells as this process begins. So here in blue, we're looking at the axon in cross section. So we've cut down in the middle, and we're looking at the cross section of the axon. And here we have a cell that is engulfing it, and this would be in the PNS. And in the PNS, we have Schwann cells as glial cells, and it engulfs the axon, and then it starts wrapping itself around that particular axon over and over again. In fact, that one Schwann cell may actually wrap itself around, and you can see these little onion-like structures beginning to form. It might wrap somewhere between 50 to 100 times. It literally winds itself around that particular axon. This will increase the membrane resistance wherever the Schwann cell has been found. And this reduces the leakage of the current out of the membrane, and that will help with the length constant or the space constant. And think of it in a lot of ways as being a form of insulation around the outside of the axon to prevent any of that signal or any of those ions from leaving, and that allows it to travel much, much further before that signal gets lost or drops to 37% of its original value. I'm hoping you're still with me, and if you are with me, just hold on a little bit more. So in the CNS, we talk about oligodendrocytes, and these oligodendrocytes actually have a number of processes. A Schwann cell just wraps individually around one segment, and that's it. One Schwann cell, one segment of an axon. Right beside it, you would have a different Schwann cell on a different segment of the axon. But the oligodendrocytes in the CNS, which are also glial cells, they myelinate in a slightly different way. So they will extend multiple processes and make contact with multiple axons in multiple areas, as you are seeing on this particular figure up here at the top. The oligodendrocyte would be in pink, and you would see that it would form these myelin sheaths. It would still wrap around multiple times, and it will allow for myelination and an increase in the membrane resistance, and as a result, an increase in the length constant as well. With me so far? All right, let's go on to the next slide. So in this process of myelination, whether it's through oligodendrocytes or it's through the Schwann cells, so either in the periphery or within the CNS, this myelination process actually leaves some very small gaps between the adjacent portions of the myelin sheath. And you can actually see that here. So the glial cell will wrap around one section, next glial cell does the next section, etc. And these small little gaps are actually where we have structures known as the nodes of Ranvier. And these nodes of Ranvier are where we have very specialized clusters of these voltage-gated sodium channels that we talked about before. There's no reason to have voltage-gated sodium channels anywhere else because this is the only place where we can see action potentials being generated. All right, so I think also to keep in mind, if you're looking at multiple sclerosis, one of the things that we know that happens in multiple sclerosis is that it is a demyelinating autoimmune disorder. So autoantibodies as well as different types of immune cells. And it's been hypothesized that both T cells as well as B cells, surprisingly, are involved in the process of demyelination. And these little myelin sheaths get lost. And you can imagine that the signal from the myelin being lost, the neuron can't communicate as effectively as it did before. And this is why we see issues in multiple sclerosis. So again, really fascinating to make use of these biological principles in the real world related to neurodegenerative conditions. So this gets into a good place to take a little bit of a break from talking about the biomechanical properties of how something spreads. And we've been looking at the RM, or the membrane resistance. We'll come back to axonal diameter in a moment. But we can also talk here, because of myelin, about two different ways in which an action potential can travel down the length of this axon. And here is a great place to talk about saltatory conduction. In myelinated axons, what you see is this jumping type of action down here at the bottom. The membrane is only exposed at the nodes. This is the only place where we see voltage-gated sodium channels. And because action potentials are only generated at these nodes, in these gap regions, that means that if you were looking from the outside or recording from the outside, it would look like the action potential is jumping from node to node instead of being spread down the length of the particular axon. And this jumping mode of conduction that we see in myelinated axons is known as saltatory conduction. Again, hoping that's clear, hoping that makes a little bit of sense to you at this point as well. So if we have an action potential on one node, the depolarizing current that is generated at that site is actually strong enough, and that was on that previous slide, that you're seeing that if we generate it here, it's actually going to generate it at different nodes simultaneously. So it's going to travel down that axon for many, many different nodes, 5 to 10 nodes. On that previous diagram, you saw 3 different nodes. And this is sufficient strength to bring all of the following nodes to the threshold potential. So it makes it easier and faster for the action potential to spread. So an action potential at one node is already preparing the successive nodes to be depolarized toward threshold at -50 to be able to generate action potentials at the next nodes almost simultaneously. And this will be a way to rapidly spread the depolarizing current between the myelinated portions. And this myelin is acting to prevent any leakage of the current across the membrane between those nodes. And we've already seen how that happens through increasing RM, or the membrane resistance. So this is really cool. And if you think a little bit about what this means biologically, so action potentials at one node may bring, not always, not in every situation, it may bring somewhere between 5 to 10 of the next nodes toward that threshold value. So they're already ready, primed, and willing to get excited. Remember, that's the only place where we see these voltage-gated sodium channels. And what happens in biology is that the most important node is the one that is the furthest away that is able to be excited, because that's going to be the depolarizing force for the next 10 nodes, and so on and so forth. So by starting off here using saltatory conduction and having a node-like structure with myelin, not only does it prevent leakage, but it actually sets up and prepares the next 10 nodes to be at threshold, ready to go to generate action potential. So starting an action potential here spreads this incredibly quickly. It's a little bit of a mind-blowing concept, isn't it? Because when I first heard that as an undergrad here at the University of Toronto, I was like, that seems so weird. Like, how does one action potential mirror and clone itself to the next few nodes? But it makes a lot of sense. There's one other really cool thing about this as well. This type of conduction in the mammalian nervous system, if I were to damage a section of an axon and some of the nodes in that axon, that means that I don't need to worry about it as much. So I could poison or block or somehow cause damage to some of these nodes, and the depolarizing current, because of the way in which this saltatory conduction works, it won't be blocked here. It won't stop here, unlike the traditional passive spread that I showed you earlier, because the next healthy patch membrane is already going to be depolarized enough. Again, a really, really cool concept if we think about it. And at this point, I would invite you to really spend some time thinking about it. It's a really, really cool concept, and it's one that we need to think about. Now, what we've done in the last 5 minutes is to take a little bit of a detour from what we were talking about. Our focus, before we got sidetracked to like nodes and saltatory conduction, was to examine the length constant, which defines like how much a signal drops. We know that one factor is the membrane resistance, and that's primarily determined by myelination, and it occurs in different ways in the CNS versus the PNS. And then we also talked about the other factor, meaning the axonal diameter, which we haven't talked about yet. So myelination definitely improves this length constant. It makes it spread much further before the signal gets lost. And then the other way to do this is to increase the axonal diameter. So if we look at two different axons, we can also increase the diameter of these two different neurons. So for example, the axonal diameter of B is going to be bigger than the axonal diameter of A. So again, this is a little bit misleading here, so apologies on that. I hadn't corrected it, but you can see the diameter is bigger here than it is over here. And that means the depolarization is going to spread further along the one that has the bigger axonal diameter, which would be down here as well. So again, just keeping in mind that the internal resistance of these are going to be inversely proportional, and this is what I think the slide is showing. And again, apologies on that. I'm going to go back and make sure that we correct that for you, or we discuss it in the discussions page as well. So unmyelinated axons, by the way, don't have this extensive wrapping on the outside. There is therefore going to be a lot of current leakage, and it slows down the conduction velocity. And the slow conduction velocities, because conduction velocity is directly related to the length constant or lambda, slow conduction velocities are related to small axonal diameters, and then low membrane resistance. And again, you can take a look at that equation that I showed you earlier. And then both voltage-gated sodium and potassium channels are mixed along the entire length of the unmyelinated axons. So it's not at these little patches. In myelinated neurons, excitable membrane patches are only found at the nodes of Ranvier, but in unmyelinated axons, voltage-gated sodium and potassium channels are found down the entire length. So we have everything going on all the time. We need to depolarize all of these different voltage-gated sodium channels. So oddly, if we were to look in the brain, or we were to take a look within the CNS, in your spinal tract, or within your brain, the majority of the axons are actually unmyelinated. But what does that actually mean? And what does that functionally mean? So these unmyelinated axons do actually have some insulation. So they are surrounded by these glial cells, and these Schwann cells that you see here, or oligodendrocytes, engulf the axon. But here, they would engulf many of these axons. And instead of wrapping around, they're just going to surround them. They're not going to go around again and again 50 to 100 times. They'll just engulf a whole bunch of these axons. We still consider them unmyelinated because the insulation isn't very thick. There is no winding process of the Schwann cells or oligodendrocytes, and we call this structure, where we have multiple axons, a Remac bundle. And that's just the name of the structure. It's the glial cell and multiple axons together that causes some myelination, but not extensive myelination. And this again is why we call it an unmyelinated axon. All right, we're almost there. There's nothing to know on this particular slide, but just again showing you the wonderful world of squid and octopi. Squids have taken a very different approach to speeding up the conduction velocity to get a signal down as fast as it can to swim and to escape. It's decided to make the axonal diameter as large as it possibly can. So that's how it speeds up its signal. We've taken the opposite approach. We are much smaller. Our axonal diameters are all the way down here on the left. Small diameter axons in comparison to squid. The squid giant axons are almost a millimeter in size. So this is 1,000 microns or a millimeter. Most of our axons are much, much smaller, but we have myelination. So look at the speed on the y-axis, and our neurons, even though they're smaller, are actually much faster overall than the squid giant axons, which are unmyelinated and are just simply large. And we need that, especially within our brain. So hopefully, if you've understood the previous 27 slides, you'll have a good idea of why myelination is so important. All right, so we're almost there, folks. Just bear with me for about 12 more slides. We're almost there. So we're getting the signal down. We're down toward the axonal terminal, and this action potential is being conducted along this membrane right till the very end of the axonal cell. And at the end of this cell, this action potential is still depolarizing patches of membrane. It can't actually go backwards. The action potential can't go back to where it started, and it can't turn around and go back to the beginning because of those refractory periods of the action potential and the voltage-gated sodium channels that we talked about in the last lecture. So if you're drawing a complete blank on refractory periods, I would invite you to go back to the end of the last lecture to review absolute and relative refractory periods and their influence on these voltage-gated sodium channels. So at the end of the axon, because it has nowhere left to go, the action potential does eventually die out, and it's only unidirectional because of this refractory period. So it cannot go backwards. It's only in this direction. So from the point that it started, it can only go in one direction. It can't actually come back in the other direction. And that's because behind where the action potential originated, it is still in the refractory period. And that means that sodium channels would still be inactivated because of that. So getting toward the end of where these neurons are connecting with each other, we get to these structures known as synapses. These connections between neuron A and the next cell are known as these synapses. And these are functional associations of a neuron with another neuron or different effector organs like a gland or a muscle cell. And we can have different types of synapses. So there can be an electrical synapse, or there can be chemical synapses. And what we're looking at here is a neuron making a synapse with a muscle cell, and we'll talk about that later on in the course. And we can also talk a little bit about these electrical synapses or electrotonic synapses. And what we're looking at here are two cells, cell on the right and the cell on the left, cell 1 and cell 2, and they're physically connected together. There's a very, very narrow gap of about 35 angstroms in between, so they're physically very close together, and then functionally they are connected to each other by means of these connexon or connexin proteins that make up these gap junctions. This will allow for ions to pass through from one cell to the other, and this type of connection is known as an electrical synapse because electrical signals carried by ions travel directly from one cell to the other through these different gap junctions. And you can actually see them under electron microscopy, all of these different types of gap junctions, very, very prominent, not just in the nervous system, but also in the cardiac system as well. So the other type of synapse would be these chemical synapses. And these chemical synapses are represented by what you are seeing down here at the bottom. In some ways, We've already talked about these chemical synapses in previous lectures. So a chemical synapse occurs when the transmitter, the neurotransmitter that is packaged into those vesicles, gets released into the extracellular space in between these adjacent cells. So the synapse is defined by the presynaptic surface, which we call the bouton, or the synaptic terminal boutons. And in the boutons, which I'll show you in a few minutes, we have these small little vesicles that I showed you earlier in lecture number 2. And then we also have the postsynaptic neuron, the next neuron over, or the next membrane over. And this space is bigger, so it's not the 35 angstroms, it's about 200 angstroms. Still very small, but the gap is actually much, much larger. And this space is actually very specialized due to the existence of the postsynaptic membrane, which has specific receptors to bind with the neurotransmitters that are going to be released. So here again, showing you that 200 angstrom gap here, and what we're looking at is a presynaptic membrane. We have these little vesicles-like structures, and these little red dots that you see here are the ones that represent the neurotransmitters. And these vesicles are basically tiny little organelles which contain these neurotransmitters and then will fuse with the membrane and will then cause release of the neurotransmitters across this 200 angstrom gap to bind to the plasma membrane of the next cell over. Associated with this, though, is this process where we have calcium. This is a very calcium-dependent process, and we'll also see that this calcium is entering, which because it's an ion, it's entering through these voltage-gated calcium channels. So here's where we start talking about those voltage-gated channels yet again. Okay, so it's been a long lecture. We're almost there, folks. And I'm hoping that you've been pausing along the way to kind of stop and make sure that you're comfortable with everything, jotting down notes where you can, or questions where you need to come back and ask. So how do these vesicles get released? So we've been talking about action potentials traveling down the axon, and we haven't really talked about how that triggers this vesicular release. And this trigger, as I showed you on that previous diagram, is calcium ions, and it's always calcium ions. It is a calcium-dependent process. But how do we get calcium to flow down its concentration gradient to get inside that neuron? So we have these voltage-gated calcium channels which get depolarized by action potentials. The membrane gets depolarized, they're voltage dependent, so they will also open in the same way. So think back, those S4 domains that we talked about, and how they're sensitive to changes across the membrane. And as a result of this depolarization, these calcium-dependent channels open, or voltage-dependent calcium channels open, calcium flows in, and this calcium diffusion into the bouton, into that intracellular space, actually triggers a cascade of different types of reactions that ultimately results in vesicle exocytosis. So whether it's the kiss and run type that we talked about previously, or the full fusion events where all the neurotransmitters are released, both of them are going to occur, and they're both calcium dependent. I hadn't mentioned that before, but it's good sort of as a reinforcement of kiss and run. So here's where you sit back and you try to remember, what details of kiss and run do you remember from previous lectures, and what makes sense to you? What can you still remember from that lecture? What was different about full fusion versus kiss and run? So again, normally these vesicles are docked. They're already ready for this fusion event. They're just waiting on calcium entry, and you can sort of see this vesicle. This is that presynaptic bouton membrane. They're ready to go. And we won't worry about all of the different snare proteins that are involved in moving and organizing how these vesicles get released. But we'll see that they are calcium dependent. So voltage-gated calcium channels are not shown here, but calcium is the key trigger to activate this process. And then we get this fusion event that allows for the contents of the vesicle to be expelled into the extracellular space. And these synapses are actually really important processing stations. And I mean that in a variety of different ways. Oftentimes we think that if we do something, or if we have a trigger for something, an action potential, for example, it's going to always result in a process like neurotransmitter release. And it turns out that that's somewhat true, but it's not always true. And it's a little bit like life if you think about it. So if you study and if everything goes right, most of the time you're going to get a really great grade on your term tests. And fingers crossed, that's going to happen for you in this course and for any other courses that you take. But the reality is, once in a while, we know that that's not the case. And chemical synapses are a lot like that. So we get these vesicles, everything is happening, action potentials are coming down, calcium is coming in, but vesicular release is probabilistic. It's not guaranteed to happen. So one single action potential only has a 10 to 90% chance of releasing that one vesicle. And what that means is the more action potentials you have invading that presynaptic membrane at the terminal, the more likely it is that vesicles are going to be released. You increase the probability of release. It's a pretty mind-blowing concept, but we can take that back to the analogy. To really enhance the odds of you doing better, it's not just studying, it's studying more often or more frequently that will help you get the better mark. Sorry to wax a little bit philosophic. Here, I think we're at the end or near the end of our slides. So here's that review slide for you to go over, review these concepts. Is there anything on this slide that doesn't make any sense to you? Is there anything that's missing? And here's a good way to go back and use this review slide and add your own information on top of it. So I tend not to read back what's on this slide, but I use this, as I'm sure Dr. Key normally would, for you to kind of add things to it, not just as a summary. So here is where I'm going to stop sharing my screen. I'm going to go back. We're going to chat a little bit before we wrap things up. It was a really long lecture today, and I'm hoping that it's okay that it was a little bit longer. So we've ended sort of that 3 lecture block. It's a lot of work, and you need to put that time and effort into it. And I want you to, again, think a little bit about how you're progressing in this course. I'm actually going to jump back to that lecture deck. So just give me 1 second, and again, apologies on this as I go and find it. And I wanted to just also just thank you for bearing with me. You're going to get into a routine. You may not have gotten into that yet. Hopefully you've set aside that time and space to be able to study on a routine basis, and you'll get into a routine. The brain is a fascinating place. I know a lot of this seems like it's just really biomechanical or it's cell biological, but we're going to get there. Just bear with me. And as we get further along in this lecture series, I'm hoping that you'll see why I love neuroscience so much, even though I wasn't a great neuroscience student, why I love it so much now. All right, so give me some feedback. Let me know what you need. Have a great day wherever you're at. Be safe, be healthy, study neuroscience, get your synapses full, and always be excited by all the different things that you're learning. All right, folks, that's it for me. Take care. See you later. See you in the next lecture.